In the male reproductive system, biology kind of runs a really steady, predictable factory floor. Millions of cells are just produced daily in this continuous, uninterrupted assembly line.
Yeah, it just never really stops.
Right, exactly.
Yeah.
But when you look at the female hormonal axis, that whole factory floor concept, it completely
Oh,
disappeared.
it goes right out the window.
It does. Biology runs this ruthless, high-stakes elimination tournament instead. I
Mhm.
mean, it builds to this dramatic peak, triggers one singular explosive event, and then, and this is the wild part, it completely reorganizes its entire cellular structure.
Yeah.
Twice a month, essentially. The whole system just resets. And when you dig into the clinical literature tracking this female hormonal clock, I mean, the sheer biological engineering of it is just staggering.
It really is. It's this intricate, highly volatile dance of hormones. Every single physiological step is sequenced to prepare for, one very specific, highly delicate outcome.
Which is what we're getting into today.
Exactly. If you picture a 28-day, almost lunar-like calendar, we can map out how this works on a physiological level. We're going to track the follicular phase, the luteinizing hormone surge, and then the luteal phase.
And connect it all to the clinic, right?
Yes. Ultimately, we're going to unpack why the timing of every single ART, protocol-assisted reproductive technology, depends entirely on clinically manipulating that exact natural rhythm.
I love it. So today, I'm stepping into the role of specialist in training. And you are our resident senior embryologist guiding us through this mission for today's deep dive. We've got a whole stack of clinical sources, medical reviews, and case studies to get through.
We do.
So let's just start at day one. The follicular phase, we have this high-stakes elimination tournament, right? How does the body even select the starting lineup?
Well, the signal actually originates up in the brain. The pituitary gland releases follicle-stimulating hormone, or FSH.
Okay, FSH.
Right? And this hormone travels through the bloodstream, down to the ovary, and essentially wakes up a cohort of primordial follicles.
And they're like tiny fluid-filled sacs.
Exactly, tiny sacs. And each one contains an immature oocyte. Now, as this specific cohort of maybe, say, 15 to 20 follicles wakes up and begins to grow in response to that rising FSH, they don't just passively sit there and absorb hormones.
They start doing their own thing.
They do. They actively participate in the feedback loop. So as they grow, they start secreting their own hormones, specifically estradiol and a protein called inhibin B.
Okay, wait, let's unpack this. I want to pause on the estradiol and inhibin B because this is where the tournament gets really competitive.
It gets ruthless.
Right, because those hormones travel back up to the brain and tell the pituitary gland to dial down the FSH production.
Right.
The growing follicles are actively shutting off the primary hormone that they require to survive.
Yeah, they're deliberately pulling up the ladder right behind them.
Which is just crazy.
It is. And as the pituitary suppresses that FSH production, the serum FSH levels begin to drop. So the smaller follicles in the cohort, the ones that, maybe didn't grow quite as fast, they suddenly find themselves starving.
Because there's not enough FSH to go around anymore.
Exactly. They cannot sustain themselves on these diminished levels. Consequently, they undergo a process called atresia.
Programmed cell death.
Yes. They physically degenerate and are just reabsorbed by the body.
But wait, why wake up an entire cohort of 15 to 20 follicles if the biological intent is to just systematically starve all but one of them?
Well, it ensures that only a highly responsive follicle makes it to ovulation. One single follicle, the dominant follicle, manages to grow slightly faster during those early days.
And because it's slightly larger?
Right. Because it's larger, it has developed a significantly higher density of FSH receptors on its surface. So it basically acquires this super sensitivity to whatever tiny fraction of FSH is left in circulation.
Oh, wow. So it hoards it.
It completely hoards the remaining biochemical resources. It survives the starvation environment that it helped create in the first place, and it continues to grow while the rest of its cohort just perishes.
It's basically a reality TV elimination show playing out in the ovary.
It really is.
But this ruthless selection process, it actually explains something we see constantly in the clinic.
Right.
Regarding diminished ovarian reserve for the listener.
Yes, absolutely.
Because the body sacrifices an entire cohort of 15 to 20 follicles every single month just to yield one dominant follicle, that finite reserve depletes significantly over time.
It drops rapidly.
Right. And as a person ages and that resting pool of primordial follicles shrinks, the remaining follicles are often, less responsive.
Which means the pituitary gland essentially has to shout louder to wake them up.
Right. which is why we look for rising basal FSH levels in a patient's blood work.
Hmm.
The brain is pumping out more and more FSH just to compensate for a less reactive ovary.
Exactly. That rising basal FSH is a direct, quantifiable marker of that biological struggle. But this brings us to a major clinical hurdle.
Okay, lay it on me.
If nature designed this elegant suppressive system to eliminate all but one follicle every month, we face a massive mathematical problem in the lab.
Oh, I see where you're going.
Right. For a successful ART cycle, we generally need a dozen or so mature oocytes to work with, but biology is actively fighting us to produce just one.
Right, here is where it gets really interesting. This is where controlled ovarian stimulation, or COS, enters the picture. Because the ART protocols in our sources are designed to deliberately override that natural dominant selection process.
Yes, they are.
And the mechanism is remarkably straightforward once you understand that negative feedback loop we just talked about. Clinicians administer exogenous FSH via daily injections.
Right, from the outside.
Right. So by pumping the body with external FSH, we artificially keep the hormone levels way above that critical survival threshold for the entire cohort.
You are totally overriding the drop in FSH. And because the hormone levels never plummet, the smaller follicles are never starved of their primary growth factor.
They get to live.
They do. Instead of one dominant winner shutting down the rest, the entire cohort of 15 or 20 follicles grows simultaneously. You are literally rescuing them from atresia.
But wait, if we are pumping the body with exogenous FSH to keep all 20 follicles alive, we've essentially disabled the ovary's natural braking system.
We have.
Because the dominant follicle usually regulates the system by suppressing the brain. But if we are manually supplying the FSH from the outside, the brain suppression doesn't even matter. Doesn't that put the patient at risk of like a massive biological pileup?
Oh, it absolutely does, which is the primary risk of ART ovarian hyperstimulation syndrome. When you disable a highly regulated negative feedback loop, you, as the clinician, have to become the regulator. You have 20 follicles all growing, all pumping out massive, massive amounts of estradiol.
Far more than the body is used to handling.
Far more. The vasculature can become leaky, fluid shifts into the abdomen, and it can become a critical medical emergency.
Man, that perfectly explains the intense monitoring schedule. When we are tracking a patient with transvaginal ultrasounds every other day and drawing blood to check serum estradiol levels, it's not just to see if the follicles are growing.
No, not at all.
We are manually driving a car where we cut the brake lines. We have to know the exact millimeter diameter of those follicles and the exact estrogen concentration, so we know precisely when to hit the manual brakes and initiate the next step.
That's a perfect analogy. You are measuring the physical and chemical output of the cohort to time the climax of the cycle, which is the trigger.
The big moment.
Yes. In a natural cycle, the system operates on negative feedback to keep things stable. But as the dominant follicle reaches its peak physical size, usually around 18 to 20 millimeters, it produces a massive, sustained peak of estrogen. And this specific high concentration of estrogen, maintained for about 2 days, it does something extraordinary. It actually flips a biochemical switch in the hypothalamus and pituitary.
It reverses the rules.
Exactly. Instead of suppressing hormones, this peak estrogen triggers a massive sudden release of luteinizing hormone.
The LH surge, the defining event of the entire cycle.
Yes. It shifts the entire axis from a negative feedback loop to a positive feedback loop. The LH surge is the singular biological signal that tells the oocyte it is finally time to resume meiosis.
Let's look really closely at that cellular mechanism for the listeners because it's wild. The oocyte has actually been arrested in prophysae, or M1, since the woman was a developing fetus.
Decades.
It has been paused in this exact state for decades. And the LH surge is the starting pistol. It's the signal to complete its first meiotic division, extrude half its chromosomes into this tiny package called a polar body, and reach M2 maturity.
But the mechanical challenge here is how that signal actually reaches the oocyte.
Right, because the oocyte itself does not possess a direct blood supply.
No, it doesn't. It is suspended in fluid inside the follicle, completely surrounded by a dense protective cloud of supporting cells called cumulus cells.
So it's insulated.
Highly insulated. When the LH surge floods the ovarian bloodstream, it doesn't touch the oocyte directly at all. It binds to receptors on the outer layers of the follicle and those cumulus cells.
So how does a chemical signal in the bloodstream physically cross through multiple layers of supporting cells into an isolated oocyte?
through a fascinating structural adaptation called gap junctions.
Oh, the tunnels.
Yes, these are actual physical tunnels made of proteins called connexins, and they connect the cumulus cells directly to the oocyte's cellular membrane.
Like little microscopic pipelines.
Exactly. Now, normally, the cumulus cells use these tunnels to pump a molecule called cyclic AMP into the oocyte, which essentially acts as a brake, keeping it arrested in that M1 stage.
Got it.
But when the LH surge hits the cumulus cells, it forces those gap junctions to close. The tunnels just collapse.
Oh, wow. So the cyclic AMP levels inside the oocyte plummet.
Right. The brake is released, and meiosis instantly resumes, driving the cell to the M2 stage.
The complexity of that cellular communication is just incredible. And the LH surge, it also triggers the physical rupture of the follicle itself, right?
It does.
Because the sources describe this process called cumulus expansion, where those same cumulus cells start producing this thick, sticky matrix of hyaluronic acid.
Yes, it gets very voluminous.
It expands the entire mass, creating physical pressure,
Yeah.
while enzymes are simultaneously weakening the follicle wall until it literally breaks open, releasing the M2 oocyte.
Now, in an ART cycle, the goal is to intervene just before that physical rupture occurs. You do not want to lose those eggs.
Right, you want to catch them before they pop.
Exactly. So clinicians administer a trigger shot, often HCG, which basically mimics the natural LH surge, and then time the retrieval exactly 36 hours later.
It's all about the timing.
It's precise. They use an ultrasound-guided needle to aspirate the fluid and manually retrieve the mature M2 oocytes before they are lost to the abdominal cavity.
And once those M2 oocytes are retrieved and handed over to your team in the embryology lab, their vulnerability is honestly terrifying.
Terrifying is the right word.
The sources went into heavy detail about the temperature sensitivity. Inside that M2 oocyte, the chromosomes are aligned on this structure called the meiotic spindle, perfectly positioned and waiting for a sperm to fertilize it. But that spindle is constructed from tiny protein tubes called microtubules.
And those microtubules are exquisitely sensitive to temperature fluctuations.
Like insanely sensitive. The lab protocols mandate a strict 37 degrees Celsius. I mean, the sources analyze the specific test tube warming devices used during retrieval.
Because you have to mimic the human body perfectly.
Right. If an oocyte cools down to even 33 or 34 degrees while just being transported across the room, those microtubules begin to depolymerize. The spindles literally dissolves and falls apart.
And the chromosomes just scatter.
It's heartbreaking. Even if you warm it back up and the spindle magically reforms, the chromosomes might not align correctly. And that leads to abnormal genetic distribution, or aneuploidy.
Which means the lab has to flawlessly replicate the exact basal body temperature at every single second of handling.
So the margin for error is essentially zero.
Absolute zero. And while the embryologists are working to protect the structural integrity of that retrieved oocyte back inside the patient's body, the biological clock just keeps running.
Right, the body doesn't stop.
No. The follicle has been emptied, but the cellular remnants left behind undergo a massive structural and biochemical shift.
So what does this all mean? The ruptured follicle transforms into an entirely new endocrine gland, right? The corpus luteum?
Yes. The cells that previously supported the oocyte, the granulosa and the Aca cells, they basically luteinize. They undergo hypertrophy, accumulating lipids, and this newly formed corpus luteum begins pumping out progesterone.
Progestation.
Exactly, the ultimate pro-pregnancy hormone.
You know, the mechanics of progesterone on the uterine lining are so fascinating when you compare it to what estrogen was doing earlier in the cycle.
How so?
Well, if we use a construction analogy, during the follicular phase, estrogen acts like the crew pouring the concrete foundation and putting up the drywall.
Right, building the structure.
It causes the endometrium to grow thick and proliferate. But a building with just walls isn't really habitable.
No, you can't live in it yet.
Progesterone acts as the electricians and the plumbers.
Oh, that perfectly illustrates the transition to the secretory phase. That's great. Because progesterone doesn't make the lining thicker, it makes it functional.
Right.
It wires the blood vessels to become highly coiled and spiral, increasing blood flow. It turns on the endometrial glands, causing them to secrete glycogen and nutrients.
It's finishing the house.
It physically transforms the proliferative tissue into a highly vascularized, secretory, receptive environment, ready to sustain an implanting embryo.
Now, if an embryo successfully implants, it releases HCG into the bloodstream. That chemical signal travels back to the ovary, acting as like a rescue beacon to keep the corpus luteum alive and pumping out progesterone to maintain the pregnancy.
Yeah.
But I want to look at the mechanism of what happens when there is no embryo.
The failsafe.
Right. If there is no rescue signal, the corpus luteum has a strict programmed lifespan of about 14 days.
And then it undergoes luteolysis, which is a highly destructive cellular breakdown.
The sources outline how this breakdown is actively mediated by reactive oxygen species. Free radicals, like superoxide and hydrogen peroxide, they just build up within the luteal cells.
They destroy it from the inside.
Yeah, these molecules damage the mitochondria and the cellular DNA, triggering apoptosis. The gland essentially just collapses in on itself, and as it physically degenerates, that massive production of progesterone and estrogen, it just crashes.
And without the structural support of those hormones, the highly coiled spiral arteries in the uterine lining constrict. The tissue becomes ischemic, it breaks down, and it sheds.
Menstruation begins.
Yes. And simultaneously, that sudden crash in estrogen and progesterone removes the negative feedback on the hypothalamus and pituitary.
The brakes are off again.
Exactly. The brain senses the low hormone levels, FSH begins to rise again, and the entire 28-day biological clock restarts to wake up a brand new cohort.
Which actually brings me to a clinical paradox that I got really stuck on while reviewing the sources, specifically the 1995 case report by Barnes et al.
Ah, yes. The synchronization problem.
It illustrates a massive complication in ART cycle synchronization.
Yeah.
Because in many ART protocols, we give the patient a downregulation medication, like a GnRH agonist.
Right.
This acts on the pituitary gland to completely shut down the body's natural LH production. We do this to prevent a premature LH surge, ensuring the patient doesn't ovulate and lose the oocytes before we can manually retrieve them.
You basically lock down the pituitary so you can control the timing of the trigger yourself.
But, and here's the paradox, if we shut down the pituitary to save the eggs, aren't we just pulling the rug out from under the corpus luteum? Because the corpus luteum relies on a baseline level of LH from the pituitary just to survive and produce progesterone. If the GnRH agonist has suppressed the pituitary, the corpus luteum is starved of its life support signal.
It will undergo premature luteolysis. And the Barnes case report highlights exactly that phenomenon. By artificially suppressing the pituitary to control the follicular phase, clinicians unintentionally destroy the luteal phase.
Wow.
The corpus luteum fails, progesterone levels crash, and the uterine lining becomes entirely inhospitable.
So we've successfully retrieved a dozen mature oocytes, fertilized them in the lab, grown beautiful blastocysts, but if we transfer an embryo back into a uterus that has no progesterone support,
it will completely fail to implant. This raises an important question, though. How do we fix the nest?
Right.
This is exactly why ART cycles that involve pituitary suppression strictly mandate exogenous luteal support. You have to manually replace the job of the failing corpus luteum.
More hormones from the outside.
Yes. Clinicians prescribe daily progesterone, either intramuscular injections or vaginal suppositories, and often supplemental estradiol. You have to manually drive the secretory phase of the endometrium, deliberately synchronizing the microscopic development of the uterine lining with the exact developmental day of the embryo you are transferring.
So if the embryo is a day 5 blastocyst, the uterine lining must have been exposed to exactly 5 days of progesterone.
The synchronization has to be absolute.
We're essentially pausing the natural biological clock, extracting the components, and then manually winding the gears back up to ensure both the embryo and the endometrium arrive at the exact same destination at the exact same moment.
It requires a fundamental understanding of every hormone, every receptor, and every single feedback loop we've discussed today.
Okay, let's consolidate those components with a rapid recall. I'll take the hot seat as the specialist in training.
Right.
Test my grasp on the mechanics, and for you listening, see if the biological logic tracks for you as well.
Okay, let's test your cycle mechanics. First rule 1. What specific hormone drives the initial recruitment of the follicle cohort, and how do the growing follicles eventually suppress it?
The pituitary releases FSH to wake the cohort. Then, the growing follicles secrete estradiol and inhibin B, which feed back to the brain to suppress FSH production, deliberately starving the smaller follicles.
Accurate. Question 2. What is the mechanism behind the LH surge, and what physical barrier must the signal cross to reach the oocyte?
Okay, so the dominant follicle produces peak estrogen, which flips the axis to a positive feedback loop, triggering the LH surge. The signal binds to cumulus cells, which then force the physical gap junctions, those connexin tunnels, to close, dropping cyclic AMP and allowing the oocyte to mature from M1 to M2.
Very good. Final question. How does an ART protocol override the one dominant follicle rule, and what critical risk does this introduce?
By administering exogenous FSH, we maintain the hormone levels above the survival threshold, rescuing the entire cohort from atresia. But because we've bypassed the negative feedback loop, we risk ovarian hyperstimulation syndrome if we don't heavily monitor the intense estrogen output.
You have a solid grasp on the underlying physiology.
The complexity of it is just so fascinating. The overarching takeaway from this deep dive is that the female hormonal cycle isn't just a background process, It is the absolute biological clock governing human reproduction.
It really is.
Every single ART protocol, whether we are overriding dominance with exogenous FSH, triggering meiotic resumption with an artificial LH surge, or rescuing an out-of-sync endometrium with luteal support, is simply a clinician attempting to mimic, pause, or manipulate this exact physiological timeline.
You just cannot intervene in human reproduction without first mastering the sequence of these biological events.
And mastering how to intervene is exactly where we are heading next. In our next deep dive, Episode 7, Clinical Decision Framework, we are going to explore how clinicians evaluate a patient's unique physiology to decide which of these ART levers to actually pull.
We will break down the clinical strategy behind choosing, say, a short protocol versus a long down-regulation protocol.
It's going to be great.
It will really bridge the gap between this theoretical hormonal physiology and actual clinical application on the floor.
But to wrap up today, I want to leave you with a final thought to mull over regarding that initial ruthless follicular selection we talked about earlier.
The elimination tournament.
Right. We know the body wakes up a cohort of 20 follicles, only to starve all but one. And we know the dominant follicle survives because it develops a higher density of FSH receptors, allowing it to hoard resources faster than its peers.
Yes.
But does biological speed equate to biological quality?
Oh, that is the question.
Right. Is the single dominant follicle that survives actually the one containing the oocyte with the most pristine, error-free genetic DNA? Or is it simply the follicle that happened to react the fastest to the shifting biochemical environment?
It's a profound unknown.
It really is.
Yeah.
Does evolution favor true genetic integrity in this selection process, or does it blindly reward sheer biochemical aggression? It's a compelling question to consider as you think about the profound and honestly often mysterious mechanics of human reproduction.